If an enzyme operates at a particular temperature optimum, the problem for physiological adaptation to the environment is to ensure that this temperature optimum actually corresponds to the environmental temperature at which the enzyme operates. Environments on Earth vary widely in prevailing temperature, ranging from frigid Arctic and Antarctic conditions to hot deserts, which present environmental temperatures that can vary over a span approaching 100 degrees Celsius. How then does the temperature-dependent kinetics of enzymes correlate with this variation? Let's use an enzyme we already know, acetylcholinesterase, to explore this question. We'll look specifically at temperature adaptation of acetylcholinesterase in fishes. Let's plot out the Michaelis constant for acetylcholinesterase versus temperature for the common trout. Trout are useful animals because they can be found inhabiting a variety of different temperature habitats, from quite cold waters to warmish. So let's first plot out the variation of the Michaelis constant with temperature for trout that have been acclimated to a temperature of 25 degrees Celsius. And here's what that plot looks like. As we expect, there's an optimum temperature for the Michaelis constant, and this is a little bit on the cool side of 25 degrees Celsius, on the order of about 22 degrees Celsius or so. However, the breadth of this curve is fairly wide, so that even at 25 degrees Celsius, the Michaelis constant is down near its optimum value. Now, let's look at the same enzyme for a fish we've been introduced to already, namely the ice fish. Remember that ice fish live in the frigid waters of the ice shelves of the Antarctic. We considered them previously because these are the fish that have no red blood cells in their blood and hence no hemoglobin. Ice fish typically live in temperatures slightly below 0 degrees Celsius. They can get away with this, of course, because salt water doesn't freeze at 0 degrees, but around -3 degrees Celsius. If we plot out the Michaelis constant curve for these fish, we find that, again, there's a temperature optimum for acetylcholinesterase, and the temperature optimum is slightly below 0 degrees Celsius. Furthermore, notice how narrow this curve is. There are two things to note about these two curves. The first is how similar the minimum Michaelis constants are for both fishes. What this means is that the acetylcholinesterase for a trout at 25 degrees Celsius works just about as well as the acetylcholinesterase for that ice fish at -2 degrees Celsius. Given the role that this enzyme plays in nervous function, this helps explain in part why ice fish can perform perfectly well at cold temperatures, in fact, as well as trout can at 25 degrees Celsius. But these animals could not wander much out of their respective ranges of temperature without beginning to run into some serious problems. If you take a trout acclimated to 25 degrees Celsius and you put it into water at -2 degrees Celsius, you'll very quickly have a dysfunctional trout. By the same token, if you take an ice fish from its normal -2 degrees Celsius and you put it into water at 25 degrees Celsius, you're likely to have a dysfunctional ice fish on your hands as well. The second noteworthy thing is the difference in breadth of these Michaelis constant curves. Let's imagine that there's a range of Michaelis constants that are tolerably close to the minimum. As long as the Michaelis constant doesn't exceed this value, the enzyme will perform tolerably well. But if the Michaelis constant exceeds this value, some difficulty might ensue. This will translate into a range of temperatures over which the enzyme performs tolerably well. The trout with its broader curve for the Michaelis constant will be able to inhabit a wider range of temperatures than will the ice fish with its narrower Michaelis constant curve. We refer to this as the thermal breadth of the enzyme. The thermal breadth of the trout acetylcholinesterase is greater than the thermal breadth of the ice fish's enzyme. There's another aspect of trout acetylcholinesterase that's worthy of mention. One of the interesting aspects of trout biology is their ability to acclimate to different temperatures. What this means is that trout that are raised around 25 degrees Celsius will be able to tolerate a range of temperatures around that value fairly well, but if you take that trout and you put it abruptly into cold water, the trout will run into trouble. However, if you gradually change the water temperature over several days, say from 25 degrees down to, say, around 2 degrees, the trout will acclimate to the cold temperature so that it continues to function well. If we look at the acetylcholinesterase kinetics of trout that are acclimated to the cold temperature in this way, we see a quite remarkable result. Specifically, the Michaelis constant curve seems to have shifted from an optimum centered around the low 20 degrees range to one now centered around a range of 2 to 5 degrees Celsius. What has happened here is not a change in the enzyme itself, but a transition to a different enzyme that catalyzes the same reaction, but is now structured to function well at cold temperatures. Thus, the trout has at least two genes that can specify two different enzymes that catalyze the conversion of acetylcholine into acetate plus choline. Different enzymes that catalyze the same reactions are known as isoenzymes or isozymes. In this case, one enzyme, isoenzyme A, is the version of acetylcholinesterase that is produced at relatively warm temperatures. Acclimation to cold temperature involves gradually replacing the A isoenzyme with a B isoenzyme, a process that takes a while to accomplish. The gene for isoenzyme B has to be activated, new proteins have to be synthesized, and these have to be slotted into the nerve terminals to replace the molecules of isoenzyme A there. This is the process of temperature acclimation. The result is that function, that is, the conversion of acetylcholine into choline plus acetate, is maintained at cold temperatures as well as it is at warm temperatures. What could be the explanation for this? To understand how temperature affects enzyme kinetics, and by extension how enzyme kinetics can be adapted to prevailing temperature, we need to refresh our memories on how protein catalysts, that is, enzymes, are built and how they work. Enzymes are, of course, proteins, and these consist of linear strings of amino acids held together by peptide linkages in a so-called primary structure. The primary structure, remember, is simply the amino acid sequence, and this is specified by a sequence code of nucleic acids, ultimately residing in DNA. These strings of amino acids are flexible, and this means that they can fold up into various higher levels of structure. These are the so-called secondary and tertiary levels of structure. When proteins fold up in this way, they form complicated three-dimensional shapes. An enzyme can catalyze reactions when one particular site on this complicated structure can bind reactants and bring them together in such a way that they can react with one another. This particular site is the so-called active site. The active site's ability to catalyze a reaction depends upon its being a particular shape, and if the shape changes, it will change the relationship between reactants that bind to the active site, and this will alter the ability of the enzyme to catalyze the reaction. This is reflected in the change of the enzyme's Michaelis constant. Temperature comes into this because it's the source of one of the complicated suite of forces that hold proteins together into a particular shape. If we look closely at the forces that are holding everything together, the protein's secondary and tertiary structure is held together by weak forces, things like van der Waals forces, electrostatic interactions, interactions between hydrophobic or hydrophilic amino acids, those sorts of things. The particular configuration a protein takes on depends upon these forces folding the protein into its so-called minimum energy state. At the same time, all of the components of the protein, amino acids, peptide linkages, and so forth, are subject to random thermal motion. This is no surprise. It's just a consequence of these things being at some temperature above thermodynamic zero, which they decidedly are. These thermal motions act to disrupt the secondary and tertiary structure of the proteins. So the particular configuration of a protein is not simply the result of these weak attractive forces between different complements of the protein. It's a balance of forces between these weak forces acting to hold the protein together and the thermal motion forces acting to disrupt them. Now you can see where temperature can have an effect on enzyme kinetics. As temperature changes, the magnitude of the random thermal motion forces increases. At the same time, temperature has a relatively small effect on the weak forces that are actually holding the protein together. Depending upon the balance of these two opposing forces, a change in temperature will cause subtle changes in the shape of the active site, and this will, in turn, bring about changes in the kinetics of the enzyme-catalyzed reaction, an effect that is evident in the temperature-dependent variation of the Michaelis constant. From here, it's fairly easy to see how temperature adaptation of enzymes can work. Let's take our Michaelis constant curves for acetylcholinesterase in trout. Remember, we have two different isoenzymes for acetylcholinesterase, a warm functioning variety that we're calling isoenzyme A, and a cold functioning variety that we're calling isoenzyme B. In our trout, these two isoenzymes are coded with different genes. Just for the sake of illustration, let's say that these correspond to two stretches of DNA on different chromosomes. If one gene was copied to another chromosome, or even to another place in the same chromosome, these two variants can mutate and be selected independently of one another. That's the origin of the different isoenzymes. When these different nucleotide sequences are translated into amino acid sequences, there will be many similarities in the identity and sequence of amino acids in the two protein varieties. In this little cartoon, these similarities are indicated by the white circles. However, there will be certain amino acids in the sequence that differ in identity and location between the A and B isoenzymes, and these are indicated by the colored circles. These slight differences in primary structure of the two isoenzymes will affect their enzyme kinetics, because these differences will alter how the proteins fold up and form their respective active sites. Evolutionary temperature adaptation then follows when gene variations that produce similar function at different temperatures are naturally selected. So, a significant part of adaptation to variation of temperature involves selection of genes that produce proteins that can sustain catalytic function at different prevailing temperatures. At one level, this is a fairly straightforward matter of natural selection on nucleotide sequences and genes that can produce amino acid sequences that can fold up into some kind of a functional architecture, that is, into a protein with a catalytically competent active site. One of the striking things about life on Earth is that life exists over nearly the entire range of temperatures that exist on the planet. The only restriction seems to be that life must occur in an environment where water is in the liquid phase. So now let's turn our attention to temperature adaptation to extreme conditions ranging from the very hot to the very cold.